name is Madeline, and as Dr. Greer said, I'm a second-year PhD student in the Nickel Lab, here to give you an overview of my project defining protective metabolic pathways in triple-negative breast cancer. Oops. So to start off, based on Canadian breast cancer statistics, we know that 1 in 8 women are diagnosed with breast cancer throughout their lifetime, which equated to around 32,000 diagnoses in 2025. What's more concerning is that despite advancements in treatments and early diagnosis, 1 in 35 women will still die due to metastatic complications. This translated to 5,400 Canadian women losing their lives last year. These statistics suggest that there's still a lot of work to be done in the breast cancer field. Clinically, breast tumors are molecularly classified into 5 subtypes, which can be seen here on the screen. These align with the expression of histopathological biomarkers used in breast cancer diagnosis. There are 2 hormone receptors, estrogen receptor or ER and the progesterone receptor or PR, and then human epidermal growth factor receptor 2 or HER2. The expression or lack thereof of these 3 receptors provides important clinical information for both clinicians and patients, including things like patient prognosis and possible treatment plans, as there are receptor targeting treatment options. We can see here that triple-negative breast cancer is the most aggressive subtype and is associated with the worst prognosis. So as its name suggests, triple-negative breast cancer, which I will refer to as TNBC, is characterized by a lack of ER, PR, and HER2 expression, accounting for between 12 to 17% of breast cancer cases. Additionally, due to the aggressive phenotype, these patients have a high risk of metastases, with over a third of patients developing distant metastases throughout the course of their disease. The lack of targetable receptor expression and high degree of metastases make TNBC very difficult to treat. And as I previously mentioned, receptor status can inform treatment decisions where the receptors can be used as drug targets. And since these are not present, treatment for TNBC patients often consists of more systemic approaches, including surgical resection, radiation, systemic cytotoxic chemotherapeutic agents, and immunotherapy. Systemic therapeutic approaches by nature are harsher and more toxic to the body. And while patients tend to respond well to therapy initially, TNBC patients often develop resistance, translating to a low overall survival and high risk of recurrence. And so with all of these challenges I presented here today, it's evident that more work is needed to improve our understanding of what might distinguish more aggressive TNBC tumors, to improve our understanding again, which may lead to better treatment to improve clinical outcomes. So what do we know? We know that previous research has focused on studying the genomic and transcriptomic landscapes of TNBC, and studies have separated TNBC tumors into 4 transcriptomic subtypes with distinct characteristics. Associated therapies have been developed based on these subtypes. However, they've had limited success, and systemic chemotherapies remain the standard of treatment for a majority of these patients. Due to the high risk of recurrence associated with TNBC and the limited success of transcriptome targeting therapeutics, evidently further research is needed. As such, current studies in our lab and among others are interested in exploring the TNBC metabolome as a way to get a more integrated understanding, because it's impacted upstream by the genome, transcriptome, and the proteome. The metabolome, which involves small molecule signaling, can be investigated to profile cellular metabolites and examine how they affect the phenotype and function of cells. So this will allow us to identify the presence of distinct metabolic signals in TNBC with the hopes of identifying tumors that are more likely to be aggressive and gain a better understanding of the TNBC landscape as a whole. Interestingly, a previous study conducted by Gong et al. Identified 3 metabolic pathway-based subtypes, MPS 1 through 3, of TNBC with distinct metabolic features. The 3 subtypes were also found to have distinct prognoses, metabolic distributions, and genetic alterations. So if you see at the top in blue there, we have MPS 1, which is the lipogenic subtype characterized by dysregulated lipid metabolism. MPS 2 in red, the glycolytic subtype characterized by dysregulated sugar and nucleotide metabolism. And finally, MPS 3 in yellow at the bottom, which is the mixed subtype, which has partial pathway dysregulation. If I can draw your attention to the survival curve here on the right, we can see that MPS 2 patients in red had the worst, significantly worse relapse-free survival compared to the other 2 groups. These tumors appear to be the worst of the worst, and this data suggests that the upregulation of sugar and nucleotide metabolism in this subtype may be linked to highly aggressive phenotypes in these tumors. As I previously mentioned, the metabolome involves small molecule signaling, and this includes sugars, nucleotides, amino acids, and lipids, and how they affect the phenotype and function of cells Metabolomics as a whole, or the study of the metabolome, provides information on tumor net effects because, as I said before, it's impacted upstream by the genome, the transcriptome, and the proteome. For the purposes of my talk today, we're going to focus on a subset of the metabolome called the lipidome, which you can see highlighted here in yellow. Before we dive into the lipidome, I want to touch briefly on metabolism for a moment, just to provide some context for the next couple of slides. So when we're talking about cell metabolism, we know that cells need fuel to function properly, depending on their needs, and there's a level of homeostasis that's maintained in a normal cell. Unlike a normal cell, a cancer cell is quite greedy, and they like to soak up all of the fuel and nutrients around to account for their needs, which allows them to rapidly proliferate and survive in the tumor microenvironment. And because of this, metabolic pathways are often dysregulated in cancers to account for their increased energy and nutrient requirements, lending to their ability to rapidly proliferate and survive in the tumor microenvironment. And if you'll recall, metabolic dysregulation is a hallmark of cancer and greatly influences both disease development and progression, as well as response to therapy. So now that we understand the context of what we're talking about, we can dive into the lipidome and how it fits into all of this. So the lipidome is the complete collection of all lipids in a cell, tissue, or organism and how their amounts and types may change over time. We can center the importance of the lipidome around four major concepts. The first being structure. As a lot of us know, lipids form cell membranes with different lipid compositions controlling things like membrane fluidity, curvature, and permeability. The second is energy. So different types of lipids like fatty acids and triacylglycerols are dense energy stores. The third being signaling. Lipids act as hormone sensors or hormones and secondary messengers, sorry. And finally, regulation. So lipids, again, can influence things like protein localization, enzyme activity, and gene expression. So essentially, if you think of the genome as the blueprint to the cell, the lipidome, sorry, is the building materials, the walls, the doors, the wiring, insulation, signaling flags of the cell. Assessing the lipidome allows us to assess how a cell is built, how it's powered, and how it communicates. And what's exciting as well is that it's highly dynamic in nature. So it's shaped by the cell's environment and disease state, reflecting what the cell is doing right now and not just what the cell's potential is. Given the metabolic reprogramming of cancer cells, exploring the TNBC lipidome will give us insights into what is building, powering, and allowing these highly aggressive cancer cells to communicate and grow. So I've mentioned a few times that cancer cells undergo metabolic reprogramming. In a normal cell, the lipidome is under tight regulation to ensure there's homeostasis and controlled proliferation. This means that fatty acid and lipid metabolism is demand-driven and tightly regulated. Fatty acid synthesis is activated only when needed for things like membrane repair, differentiation, or signaling, and fatty acid levels are kept within a narrow range. Fatty acids are also efficiently incorporated into membrane lipids and stored when in excess. So that means that membrane lipids are produced to support membrane integrity, proper signaling, and barrier function. And because of all of this, proliferation is controlled and occurs when necessary. And so you can think of lipid metabolism serving as a function of the cell first and growth second. In cancer cells, lipid metabolism is growth-driven first, leading to increased and uncontrolled proliferation. To accommodate the proliferative needs of the cell, fatty acid synthesis is constitutively upregulated, leading to the accumulation of excess fatty acids, as well as increased membrane lipid biosynthesis. This increased membrane lipid production is what allows for more rapid membrane expansion, as well as altered membrane composition supporting oncogenic signaling. Proliferation is left unchecked because lipid metabolism is hijacked to fuel growth and survival. So what are these fatty acids and lipids? How do they come together to form the membrane? And why are they important? Very briefly, fatty acids, which by themselves cannot form membranes, are activated to form fatty acyl-CoA, and this step makes it ready for lipid synthesis. In step 2, the fatty acids are attached to a backbone molecule. And then finally, in step 3, these newly formed lipids are amphipathic. So that means that they contain both a hydrophilic head and a hydrophobic fatty acid tail. And so they spontaneously self-assemble into the lipid bilayer, with the fatty acid tails facing inward and the polar head groups facing outward. Finally, fatty acid properties determine membrane behavior. And this becomes really important where longer fatty acid chains make membranes thicker. Saturated or straight-chained fatty acids will decrease membrane fluidity because they're more rigid. And unsaturated fatty acids, which contain a double bond and have a kink in their chain there, will increase flexibility. And so this is why cells carefully regulate fatty acid synthesis, elongation, and desaturation. And this is important in cancer cells because they have to precisely regulate their own fatty acid chain length and saturation because they want to create membranes that are stable enough for signaling, but flexible enough to survive rapid growth and stress. Before we dive into fatty acid synthesis and lipid membrane formation and how it's regulated, I wanted to touch on the transcription factor peroxisome proliferator activated receptor gamma, or PPR gamma. PPR gamma is a ligand-activated transcription factor belonging to the nuclear receptor superfamily, and it's expressed in mammary gland epithelial and associated stromal cells. When endogenous ligands like fatty acid metabolites, keyword fatty acid that we've been talking about, and thiazolidinediones such as rosiglitazone bind, they cause a conformational change resulting in increased transcription of target genes. And this is namely genes involved in adipocyte differentiation, sugar and lipid metabolism, which is really exciting and we'll be talking about soon. And additionally, previous work in our lab has shown that it also plays a key role in suppressing breast tumor progression, which we will also be discussing in the coming slides. So now that I've introduced you to PPR gamma, I want to talk a little bit about how it functions to tightly regulate lipid metabolism. Transcriptional regulation, generally speaking, helps determine what metabolic machinery this gets built by the cell. PPR gamma specifically acts as a lipid-sensing nuclear receptor, and when activated, it changes gene expression programs related to fatty acid handling. PPR gamma doesn't just turn on fatty acid synthesis, it couples it with safe handling. So the newly made fatty acids are directed into appropriate pathways, and that includes having a balance between the synthesis of fatty acids with fatty acid uptake and storage, and preventing this accumulation of free fatty acids. On the right, we just have some major outcomes of PPR gamma activation in the normal cell. So it supports lipogenic programming, promoting expression of enzymes involved in fatty acid synthesis and lipid assembly, controls fatty acid availability for membranes, crosstalks with other major lipogenic transcription factors, and redirects fatty acids into storage as neutral lipids. And so the key takeaway is that PPR gamma makes fatty acid synthesis safe and coordinated and helps maintain that homeostasis I was talking about in normal cells earlier. I also wanted to note that PPR gamma expression loss can occur in cancer cells, and this is often associated with a more aggressive phenotype. And if you recall, free fatty acids are what get integrated into those lipid membranes I was talking about earlier. So you can kind of imagine how PPR gamma may help regulate lipid metabolism in a cancer cell and why a cancer cell may not appreciate PPR gamma's hard work. As I mentioned a slide ago, PPR gamma has been found to suppress breast tumor progression. So Liz, a previous student in our lab, began investigating the impact of expressing and activating PPR gamma on TNBC tumors using an in vivo xenograft model. For those who are unfamiliar with the xenograft model, Liz injected MDA-MB231 cells derived from human TNBC orthotopically into the mammary fat pad of immunocompromised mice. Tumor growth was then monitored over time, and in this case, the study was a 6-week study. For the sake of time and space, I've only included the 1st and 6-week images here. In the first panel at the top here, we can see tumors formed with the parental cell line, which does not express PPR gamma at weeks 1 and 6. So evidently you can see from week 1 to 6, there's a lot of tumor growth and some spread suggestive of metastasis up towards the lungs. The same was done using these parental cells again. However, the mice were treated with rosiglitazone, or Rosie, the PPR gamma ligand. So seemingly smaller tumors with less lung metastasis. The same was done using these cells transduced with a PPR gamma wild type plasmid. So we're able to induce the expression of PPR gamma in these tumors. And then finally, we see the most significant decrease in tumor size in the PPR gamma expressing and activated tumors compared to controls. We can show you this graphically as well, where we observe a significant decrease in primary mammary tumor volume when PPR gamma is expressed and activated. With this, we wanted to then metabolically profile these tumors to assess PPR gamma dependent and independent effects on tumor metabolism. This slide just takes a moment to load. There we go. To metabolically profile these tumors, we use what is known as desorption electrospray ionization mass spectrometry imaging, or DESI-MSI, or DESI, sorry. DESI is a non-destructive mass spectrometry imaging technique that allows for the detection of small molecule metabolites and lipids in tissue samples at atmospheric pressure and ambient temperature. In DESI, if I can direct your attention to this little figure up here, in DESI, high velocity ionized solvent droplets desorb the analytes directly from the sample surface into gaseous droplets that are delivered to the mass spec inlet over here, where the mass to charge ratios are analyzed. I want to point out that we conduct DESI at a resolution of 100 by 100 microns, and that represents 1 pixel. Each pixel has its own spectrum. Because DESI also maintains the spatial resolution of its data, producing these 2D heat maps, it allows the location of the detected ions to be analyzed in comparison to its pathologically annotated tumor and non-tumor regions. This means that we know where the data is present on any given part of the slide. Next, I'm going to briefly go over my data analysis pipeline because I think in order to understand the results I'm going to present, we need to understand the mass amounts of data we're working with. So first, the tumor samples are annotated by a pathologist, which allows us to visualize the pathological zones. In our case here, we're looking at tumor outlined in red, necrotic in orange, and then the unlabeled is the remaining non-tumor. I then take this pathology annotated image and overlay it with the DESI-derived heat map. And the rest of this analysis is conducted in Mass Vision, which was created by one of our collaborators. So once we overlay these images, we can then use the software that he developed to plot regions of interest, or ROIs, that correspond to each pathological zone. And if you'll remember, the last slide I mentioned DESI in 100 by 100 micron pixels. Each of these pixels contains their own spectra. And when we plot an ROI, I then merge 9 of the pixels together to form one spectra per ROI. The data that I pull out is then normalized, binned and filtered, and then we can apply statistics to it. So the DESI was used to analyze the tumors from the xenograph study that I mentioned earlier, and principal component and linear discriminant analysis were run to generate the plot you see here. Each dot on this plot is an average of 9 of the pixels discussed previously. There are a few important features I wanted to point out here. The first is that the sample is clustered based on tissue type. So to the left over here, we have the tumor ROIs, and to the right over here, we have the non-tumor ROIs, highlighting that the model was able to differentiate between tumor and non-tumor tissues. What's even more exciting is we see that the sample is clustered by xenograft group. So at the bottom here, we have the parental tumors. In the middle here, we have the parental tumors with Rosie, and then the PPAR gamma wild type tumors. And at the very far end over here, PPAR gamma wild type treated with Rosie tumors at the far side there. So this is really exciting because this shows that we have determined metabolic patterns that are able to identify differences not only between tumor and non-tumor regions, but also between the treatment groups. One of the ions found to be present in these tumors was oleic acid. This is a monounsaturated fatty acid, meaning that it has one double bond, that has been shown to promote the growth of highly metastatic breast tumors. As you can see here, the expression and activation of PPAR gamma significantly decreased the presence of oleic acid in these tumors. If we're comparing the red bar to the green bar at the end there. So coming back to what we know about cancer cell metabolism, cancer cells like to have these desaturated fatty acids because they prefer, or unsaturated, sorry, fatty acids, because they prefer to have more fluid membranes for movement. And so this could be an example of PPAR gamma shutting down a fatty acid source that may be fueling growth and metastasis. So what do we know? We know that cancer cells exhibit uncontrolled cell proliferation, increased fatty acid synthesis, and increased membrane lipid biosynthesis. And we also know that when PPAR gamma is activated, it controls fatty acid availability for membranes, limits lipid availability for growth, and suppresses breast tumor progression. But what we don't know is, does PPAR gamma decrease the growth and spread of TNBC through metabolic reprogramming? This leads me to my hypothesis, which is that exact question just reworded. And I aim to look at this through 3 separate aims. The first is to assess the effects of activating PPAR gamma signaling on TNBC in vivo using a xenograft model and human representative TNBC cell lines. The second is to define DESI profiles of my in vivo TNBC xenografts and correlate findings with the tumor outcomes. And finally, I hope to characterize the in vitro phenotypic effects of activating PPAR gamma signaling in my metabolically distinct TNBC subtypes. So before we dive into the methods and the results, I want to briefly talk about the cell lines that I will be working with. So here I have a table outlining some key characteristics of the HCC 38, HCC 1937, and MDA MB 231 cells, which I'll refer to as 38s, 1937s, and 231s respectively from here on out. What's important to note is that all three of these cell lines endogenously express very low levels of PPAR gamma. I also want to point out, if you'll remember earlier when I mentioned the MPS TMBC subtypes, the cell lines here are representative of each of the three subtypes. So now that we know what we're working with, we can dive into my first aim, defining metabolic changes occurring in preclinical TMBC xenograft models. The cell lines I mentioned before, I want to point out, were transduced with a WPI luciferase reporter plasmid and DOX-inducible PPAR gamma wild type vector. So these allow us to first track the cells in vivo, and then second, induce the expression of PPAR gamma in these cells. I then injected the cells orthotopically into the fat pad of immunocompromised mice. And these are the four groups here, similar to Liz's study. So the first being the parental cells that don't express PPAR gamma supplemented with a normal chow. The second being the parental cells supplemented with the PPAR gamma ligand ROSI. The third group being the PPAR gamma wild type group, where PPAR gamma expression is induced, and they're supplemented with normal chow. And then the final group is the PPAR gamma wild type group supplemented with ROSI. I then use luminescent imaging to monitor tumor growth in these mice for up to 6 weeks, at which point they are necropsied and the tissues are collected and frozen. I wanted to start by increasing the sample size of Liz's previous study. So I started with the 231s, and we now have an N of 10 per treatment group for a total of 40 mice. We can see qualitatively here the differences in tumor growth over the course of 6 weeks in the respective groups, with the most growth occurring in the parental group at the top there, and the least amount of growth occurring in the PPAR gamma wild type with ROSI group down at the bottom. I can also show you this quantitatively by plotting the average area by average density of the luminescence. And going from top to bottom, we have the parental tumors in pink, the parental tumors treated with ROSI in blue following that, and then the PPAR gamma wild type tumors in green, and finally the PPAR gamma wild type tumors treated with ROSI in orange. And this is reflective of the representative mouse images. And we see that the expression and activation of PPAR gamma displays decreased luminescence, which is further decreased when treated with ROSI. At endpoint, I do measure tumor volumes as well, and we can see here, again, the significant decrease in tumor volume when PPAR gamma is expressed, comparing this pink bar here to the green bar down here. And we get even further significant decrease when PPAR gamma is expressed and activated, looking at this orange bar down at the bottom. So far for my first aim, I have generated the tumors for the 231s and the 1937s. All the cells are ready to go, and my 38s xenograft is in progress, and the analysis is pending for that. I then went on to assess the metabolic profiles of the xenograft tumors generated in AIM-1. So the tissue samples were sectioned and run on the DESI, and then that same slide that we DESI'd was then stained and annotated by a pathologist so that I can then conduct data and pathway analysis. Following the analysis framework that I described earlier, I was able to train a model and generate this PCA-LDA that you see here. This is kind of a lot to look at, so we'll walk through it kind of slowly. Just for a reminder, I have 4 different treatment groups with 3 pathological zones. So over here on the right-hand side, in green, whoopsie daisies, green, yellow, and orange, we have the tumor, non-tumor, and necrotic region of the parental group. Slightly in front, we have the same, the tumor, non-tumor, and necrotic region for the parental with ROSI group. On the bottom left here, again, tumor, non-tumor, and necrotic for the PPAR gamma wild type group. And on the top here, we have the PPAR gamma wild type group with ROSI, with the tumor, non-tumor, and the necrotic tucked in behind there. What's really is exciting is we get this really nice separation of both of the groups that are expressing PPAR gamma. The parentals kind of are more closely associated together. And what I also think is interesting about this PCA-LDA is that the tumor regions and the necrotic regions across the board for all 3 groups group more closely together than the non-tumor regions. So we're getting that nice separation. Out of the 15,243 ROIs, 15,061 were correctly predicted, while 182 were incorrectly predicted, giving us a balanced accuracy of 97.56%. So essentially what this means is that the model can predict ROIs belonging to 3 pathological zones within 4 treatment groups, 97.56% of the time. For this analysis, I started with a subset of my data, looking at ions significantly elevated in the parental group compared to the 3 other groups in the 50 to 500 mass to charge range in order to assess the impacts of PPAR gamma expression and activation on metabolite abundance compared to controls. I ran a t-test to identify ions significantly elevated in the parental tumor regions and then sorted these by p-value, pulling out the 25 most significantly elevated ions. Unfortunately, we do not have time to go through all 25, so I've put 3 up here for you that really stood out to me, and this includes arachidic acid, gondoic acid, and cirrhotic acid. Just to orient you to this first graph here of arachidic acid, the pink represents the parental group, the blue is the parental treated with Rosie, green is the PPAR gamma wild type group, and finally orange is the PPAR gamma wild type group treated with Rosie. There's a lot to look at here, so I'm just going to direct your attention to these bars here. We see a significant, pardon me, we see a significant decrease in ion abundance with Rosie treatment alone, comparing the pink and the blue bars here. And this is exciting because it highlights the systemic effects of PPAR gamma, as well as the potential crosstalk occurring between tumor and stromal cells. Further, we see a significant decrease when PPAR gamma is expressed, comparing the pink bar to the green bar here, as well as what's most exciting is that there is a significant difference between parental tumors and PPAR gamma wild type treated with Rosie tumors here, highlighting that the expression and activation of PPAR gamma is what most significantly decreases the abundance of these ions. We notice the same trend in the second ion here, which is gondoic acid. Gondoic acid is a really exciting ion because published work from our lab recently showed that elevated levels of gondoic acid are associated with increased lymphovascular invasion in colorectal cancer. And that's a prognostic feature that's associated with poor outcomes. So this is interesting because it could reflect a more aggressive phenotype in the untreated tumors here. And finally, we observe the same trend in the third ion as well. We can then visualize the ions I just talked about in relation to the pathologically annotated slides. So on the left there, we have representative images of the parental tumor sample. And on the right, we have a representative image of the PPAR gamma wild type with Rosie sample. The left top image of each of the panels is the H&E image, where tumor again is outlined in red, necrotic in orange, and the rest is the non-tumor region or the residual mammary gland from the mouse. You can see here how the DESI image would really nicely overlap with the pathologically annotated slides. And you can also see that there's varying ion abundance across the tissues with the ions represented here. And they're more abundant seemingly in the tumor tissue than in the non-tumor regions. And what's really cool is when you look at the parental image for gondoic acid, so this far bottom left here, you see that there's a high abundance of this ion in the tumor region, but a stark contrast. It's a lot darker in the non-tumor region. However, looking at that same ion in this PPAR gamma wild type Rosie group, you don't see as stark of a contrast. It seems a little more uniform throughout, which I just thought was an interesting thing to point out. So this led me to wanting to take a look at what I found in my original search and apply it to the non-tumor regions of the parental and PPAR gamma wild type with Rosie groups to more profoundly assess what's going on in these non-tumor regions. And looking at the bar graph for this ion here, so we're starting with arachidic again, we see what I showed previously, the significant difference between the parental tumor and the PPAR gamma wild type tumor treated with Rosie. And what's interesting is that that's reflected in the non-tumor regions as well. So in light pink here, we have the non-tumor region of the parental group, and then the non-tumor region of the PPAR gamma wild type with Rosie group. And there's a significant decrease in the abundance of this ion. And this is the same for gondoic acid as well as cirrhotic acid. And again, this just really highlights that PPAR gamma expression and activation, there's a systemic effect happening here. So why do these ions matter and why should we care about them? If you remember earlier, I talked about in cancer cells, lipid metabolism is growth driven, leading to this increased and uncontrolled proliferation. To accommodate for the proliferative needs, fatty acid synthesis is upregulated, leading to that accumulation of excess fatty acids and increased membrane lipid biosynthesis. So if we take a look at this pathway, acetyl-CoA will be the original carbon source for fatty acid synthesis, and it gets converted to malonyl-CoA, which then, with fatty acid synthase and NADPH, will be able to build a fatty acid chain in 2-carbon increments. And after multiple cycles of this, we wind up with a 16-carbon saturated fatty acid called palmitic acid. And this is the jumping off point for a lot of different fatty acids, and it's subject to a lot of further modification. So elongase enzymes will take palmitic acid and elongate it further into stearic acid and beyond. And at the same time, desaturase enzymes can introduce double bonds, creating unsaturated fatty acids, either monounsaturated if it's one double bond or polyunsaturated if it's 2 or more. And this, again, remember, increases membrane flexibility. So together, fatty acid synthesis, elongation, and desaturation generate more long-chain fatty acids that then get incorporated into the phospholipids, directly shaping membrane structure and function. And this is key in cancer cells because it allows them to have this constant lipid supply to fuel their proliferation and growth. And the fatty acids generated here, again, go back into membrane assembly. And so how does this relate to the ions that I found? If we look more closely at this pathway here, the ions that I identified here are members of the saturated and monounsaturated fatty acid synthesis and elongation. So we've got arachidic at the top here, cirrhotic acid down towards the bottom, and gondoic acid to the right here. So this pathway is really important because, as I mentioned, de novo fatty acid synthesis is occurring in tumor tissues, leading to increased concentrations of palmitic acid, which is further converted into all of these saturated and monounsaturated fatty acids. And gondoic acid, similar to how I mentioned in oleic earlier, is evidence of desaturation to form unsaturated fatty acids, which are crucial for cancer cell membrane fluidity. All in all, these fatty acids go on to be the building blocks for the more complex glycerophospholipids that get used to form membranes that are, again, stable enough for signaling, but flexible enough to survive rapid proliferation and stress. And so this is an example of how PPAR gamma may be rerouting or shutting down the cancer cell's fuel stores. Just in quick summary of my second aim, because I'm still waiting on the tumors for the 38s, everything is pending for that cell line. For the 1937s, I'm in the progress and will be conducting DESI in the coming months. And then for the 231s, I've completed the DESI run and I'm still working on some further analysis. Finally, we have my third aim, where the same cell lines that I outlined earlier were subjected to the assay conditions and treatments listed here, which I'll get into in a moment. And then I conducted invasion and proliferation assays as well as protein analysis. And then I plan to correlate these outcomes with my in vivo studies. For sake of time, again, I'm only going to present my 231 invasion results. Briefly, these cells were exposed to the following conditions again, seated on top of a matrigel layer in a 96-well transwell plate and allowed to invade through the matrigel towards a filter for 6 hours. I apologize, my graph says 24, it's supposed to be 6. They were fixed and stained and then visualized, and I conducted my analysis. Just along the bottom here, I have some representative images that just show you visually what that staining looks like and what the difference can look like. It's a little harder to tell, but the difference in amount of cells invaded is. So before diving into the results, I just want to quickly touch on the treatments and conditions. So doxycycline or dox is used to induce the expression of PPAR gamma in the cells. Rosiglitazone again, or Rosi, is the PPAR gamma activating ligand. And then next we have the dysregulated lipids and glycosylated end products, which are meant to mimic a metabolically dysregulated environment. So this invasion was conducted in triplicate for each treatment 3 independent times and reported as invaded cells with standard error. To orient you, on the left here, we have the parental cells underlined with 231P, and then on the right here we have the PPAR gamma wild type groups. The black and orange are the cells only and vehicle controls respectively. In green is treatment with Rosi. In dark blue, dark red, and dark purple, which you can see
Use your pointer on.
Yeah. Oh, yeah, that's a great idea. Dark blue, dark red, and the purple over here. Those are the dysregulated lipids, glycosylated end products, and then a combination of the two, respectively. And then the lighter colors, so the light blue, the light pink, and the light purple, correspond to their darker counterparts, however, have been treated with rosy. We can see here, unexpectedly, there was no significant increase in invasion under the metabolically dysregulated conditions. So comparing these darker colored bars here to this orange bar, there was no significant difference or elevation in invasion. However, regardless of the metabolic condition the cells were exposed to, PPAR gamma activation still significantly decreased 231 cell invasion. All right, and so these are some of the assays that I hope to complete in the coming months. Completing this assay for the 38s and the 1937s, and completing my proliferations to an N of 3, and then protein analysis depending on what I find in my DESI analyses. Some future directions, just summing up kind of the summary for all of my aims and the future studies that I hope to conduct in the coming months or year. And with that, I would really like to thank all the members of my lab. They are the most noble of gases, as you can see, and all of our collaborators. Special thank you to Chris, always, my supervisor, and my thesis advisory committee members, and Dr. Greer, of course, our program coordinator. Thank you. I'm happy to take any questions. Thank you. I'm ready.
Go ahead. Who's first? Not me.
Not all at once.
I'll start then. You guys think of your questions. So, very cool analysis, your PCA analysis. One of the things that maybe I haven't thought of this before, but it certainly clicks now is the non-tumor area in all those cohorts are not different. So the tumor seems to be influencing your analytes in a way that makes them closer to the tumor. The non-tumor is closer to the tumor than that non-tumor is to one of the other types of tumors. Do you have an explanation for that?
Sorry, I might just go to the slide.
Yeah.
Because you're talking about this here, right?
Yeah, so the non-tumor area is always close to the tumor, but the non-tumor areas for those different types of tumors are not anywhere near one another. But they're all non-tumor areas, right?
Oh, I see what you're saying. I think it's difficult to say specifically looking at a 2D plot because I don't know what this looks like in 3D space. So they may, in a 3D space, they may cluster closer together or similarly to what I showed earlier, clustering kind of to the left and the right of that Z axis there in that earlier frame. So that could be a reason that we're not seeing that. An explanation for why that might be. I mean, there's a lot of like crosstalk obviously between tumors and the stromal cells. And so that could be a reason that they're not like as closely related because when you have, for example, in the PPAR gamma expressing an activated group, there's going to be different messaging between the tumor and the stromal cells than perhaps in the parental groups. That could be a reason, but I'm not really.
Sure, I don't know how many analytes there are in this analysis, probably what, hundreds, thousands?
Thousands, yeah.
So the analytes are, I don't think that they're necessarily diffusing away from the tumors.
No.
They're presumably still associated with the cells in that area. But it's interesting that there may be some kind of paracrine interaction of the tumor with the non-tumor stroma surrounding it. Actually.
Yeah, and I think too, it's something I can look into. Like I could do a more targeted analysis on the non-tumor regions of these samples to investigate that. It's just not something that I've done yet.
One other question, David?
I guess. So the first thing is it possible that you would see these lipid changes in fluid samples, biopsy samples, so microvesicles that are circulating, or maybe hematopoietic cells that are circulating? Would you expect to see lipid changes in the membranes of those particles, those cells?
That's an interesting question. I can't say that I've thought of it before.
So these metabolites diffuse out to some extent. I mean, you use the word systemic
Mhm.
involvement at some point. So I'm just wondering whether you could get access other than just looking at the tissue itself, the cancer tissue.
Yeah, I mean, if I had to hypothesize, I would say you would be able to detect, like my mind goes immediately to like type 2 diabetic patients and how like in that metabolically dysregulated environment, there's like a high level of lipids and sugars, and PPAR gamma functions really well to help with insulin sensitivity, for example. So I don't see why you wouldn't be able to pick up on these metabolites in the blood. I am not sure about like the sensitivity or like understanding the specificity of whether it comes from the tumor or not is something you'd be able to tell from a liquid sample. But those are definitely things to think about.
So you did a really nice job showing the pathway of how your lipids are being created with the increase in size and everything. Do you think you would see similar profiles in like an area where you're getting naturally occurring growth, like maybe in wound repair or like embryogenesis or something like that? Has anybody looked at that before?
That's a great question. I haven't come across that in my reading, and it could be that I just need to broaden my scope a little bit to embryogenesis. I would presume that again, when I was talking about kind of cells fueling themselves for their like needs and growth not being first. I think if a primary mechanism of a cell is growth, then I think there would be some similar signals, but I don't know if I would hypothesize that they would be to the level of a cancer cell, if that makes sense.
And just to follow up, I mean, a lot of my understanding of what from what you were saying is a lot of what we're seeing is the cells are creating new membranes, and that's the main driving source that you're picking up when you're viewing the mass spec. Is that correct? Or is this just within the cell and in the machinery of it actually just metabolizing?
Yeah, so this is like a proposed mechanism, so I can't say for certain that this is what's happening. When we pull out the metabolites and we do their identifications, they're putative IDs, and so I can't definitively say like this is the pathway that is going down, but just with what popped up and the amount of different members of this pathway, some of the other top 25 ions were more members of this saturated and monounsaturated fatty acid pathway. That was kind of like the logical flow that I followed. And with the literature, it was kind of matching up and aligning that these fatty acids are really present in rapidly proliferating cells. I don't know if that answers your question fully.
You just can't identify what their end goal is going to be.
Right, yes. But I can take what I've found and I can look down further at the pathway and do more targeted analysis. So this was an exploratory way of looking at things, and I was letting the data kind of speak to me and tell me what it found was important. And from here, I can take those ions and I can explore those pathways because I can identify what their possible ionization would be and their mass to charge ratio. And I can sift through my data to look for those ions and graph them and see how, like, what the levels look like. Like, for example, with palmitic acid, which is at the top of the chain, I would expect to see a lot of that as well, but perhaps not as because it's flowing through that pathway so quickly. So something that I'm hoping to do is to look at some of the longer chain fatty acids in that pathway and see if they're popping up as well.
Michael.
Do you see any differences in endosomal function when you have differences in the types of fatty acids that are present? Is that something you can add maybe?
It's not something that I've looked into, but that's a really great suggestion. I could definitely look into that. Thank you. Thank you.
The patients that have like perigamma extubation, they respond better to treatment, like the triple negative cancer?
Oh, there's epidemiological studies that show that patients on type because rosiglitazone is a second line type 2 anti-diabetic drug, and there's studies that show that patients who are taking a PPAR gamma activating drug do have a better outcome.
And do you compare like your drugs to metabolic profiles from patients to see if you have like similar?
I haven't done that yet. I think that's something that would be a great future direction for this project, and I hope that if I don't get to do it, somebody does get to compare it to patient samples, because I think that ultimately, right, that's the most clinically relevant sample, is to get to that, the human samples. But yeah, great question.
So just two more little questions.
Sure.
Nice, demonstration of all the metabolic pathways that are involved in the production. I'm curious about the enzymes involved in driving all of those conversions. And so do you know, if any of those enzymes are actually regulated by PPAR gamma? And you start to put together some kind of a rationale for how PPAR gamma regulation is influencing and stuff.
Yes, you're reading my mind. I actually was just talking with Chris the other day about looking at some of the enzymes and doing some reading on how they relate to PPAR gamma. I haven't nailed down, in my reading, I haven't nailed it down quite yet. But that is my next step, because I think it would be interesting to probe for these enzymes or look at enzymatic activity as like a future direction. Yeah. Mm-hmm.
And the last question, have you thought of, or have you actually profiled the lipidomic patterns of just these cells, not tumors, but cells? And can you validate that some of the same differences in these lipids are present in cultured cells?
Sorry, yeah. So I haven't done that yet, but that can be done. And that's also something that we're working on, is creating these cell pellet blocks as kind of a pseudo control for, like, on the same slide. So unfortunately for this study, I didn't have that, but that is something that we're working on developing.
Because,
Yeah.
there's the issue of what flies, what comes off with the DESI versus what's there, the
Right.
totality of it.
Yeah,
And if you know what you're looking for, you can just sit on those analyzed.
absolutely. Yes.
What's
Yeah,
going on
Evelyn.
there?
Any other dimensionality reduction or classification tools?
That's a great question. Not yet, but I will be. Yeah. And I might send you an email, actually. Yeah. I used to ask Evelyn a lot of questions in 828. I think
Any other questions?
there's a question from Monty on Zoom. He says, can any correlation be drawn between your findings within each metabolic subtype of TNBC and where they fall within the transcriptional subtype classification? Just wondering if any transcriptional signatures can explain metabolic readouts. So that's a great question, Monty. There, in the study that was conducted where the MPS subtypes were identified, they did go back and try to correlate the patients into their respective transcriptomic subtypes. However, there was no uniformity with which MPS subtype they fell into, depending on their transcriptomic subtypes. I don't know if that answers your question.
Some signal. Nothing, no signal coming. Nope. Okay, if no other questions, then join me in thanking Maddie for a wonderful.
Thanks, Scott.